Has it been started?
Yes.
Okay. Hi everyone, and welcome to the IBH webinars. And my name is Yunqing from the James Hutton Institute. Sorry about the delay. We have some technical issues with the joining link. And thanks everyone who has joined this webinar so far. And today we have the pleasure to have Dr. Gert Hansel from Heinrich Heine University in Düsseldorf. He currently leads the Center for Plant Genome Engineering there. And Dr. Hansel is a molecular biologist by training. He holds a PhD from the University of Braunschweig in Germany. His research interests include agrobacterium-mediated gene transfer methods for crop plants such as barley, sunflower, and amaranth, as well as targeted genome modifications using custom endonucleases, including CRISPR-Cas. He's also interested in molecular farming and improving the photosynthesis and the stress resilience of crop plants. He's a contributing investigator for the Cluster of Excellence in Plant Science. He's also a member of the management board of the German Plant Biotechnology Society and the convener of the Association of Applied Biologists Specialist Group Plant Ed, Genome Editing in Plants. And today, Dr. Gertz will talk about precision breed plant editing. And now the floor is yours, Dr. Gertz.
Many thanks for this kind introduction. And I would like to start with thanking Malcolm and the organizers at the International Barley Hub for giving me the opportunity to talk here and present some of our data. So you heard already where I'm from and what's my main interest, and the title you have also right here. So that's why I will directly jump into the topic. So to give you a brief outline, after an introduction, I will start with barley as a case study in crop domestication, as I have a more than 25-year experience working on barley, and then we'll quickly introduce CRISPR as a domestication tool. We'll talk about challenges for the development for new grain, cereal grains, and then from barley to amaranth to make the conceptual leap. So to start with, and you all are aware of those demands. So we have an increase in the world population. So during 1 minute, so the population will grow by 120 people. By next week, there will be another 1.2 million people. And by 2050, we expect to have an increase by 2 billion people. And all of this, of course, need a proper nutrition. And so this then make the demand and the challenges for the farming under a changing environment. And here on this slide, I summarized numbers. So while in 1950, so the farmers had to produce 0.4 billion tons of cereals. So in 2000, it was already nearly 2 billion tons. And to feed all these people, which I just mentioned on the slide before, we would need another increase of another 60% to feed all this rapidly growing population, which is, of course, a huge increase which is needed. And if you look how those people are fed these days, so you will see that the world nutrition relies heavily on grains, as illustrated here nicely on this picture. So, and if you then look into what we eat, so here, these are a bit outdated numbers, but I liked the image. So here in nearly 2000, 1999, wheat was dominating before corn, maize, rice, and some other grains. And I looked into recent numbers, which would change the position number 1 and 2, more or less. So corn is now the main number, the main grain for the world's nutrition, followed by rice and wheat, which are more or less equal. And we have here all the other ones. And so if you then look into this composition, so you have here the oilseed grains, then the Fabaceae family, the legumes, you have the Boraceae family, And you have the pseudocereals, and I highlighted here already two of the ones which I will talk today. So I will talk about barley, and then we'll also talk about amaranth. So you are all aware of the domestication of plants, which took place around 10,000 years ago. And last year I had a chance to attend two conferences in Egypt, where it's very close to the cradle of domestication, which you are all aware of, the Fertile Crescents here illustrated in this dark green. And so here also the domestication of barley started some 10,000 years ago. And if you look what traits these ancient farmers were looking at, so these are summarized here on this slide. So there are things which are, of course, very common, like increased seed number and reduced seed shattering, as you know, if you look into the white species. So these typically tend to spread their seeds in order to ensure the further distribution and growth, but while this is, of course, not in the interest of the farmers. So seed shattering is one of the main domestication traits. Yield is, of course, always important. So like increased seed number. As height, plant height has also an influence on the harvesting and also resistance or tolerance to lodging. So this was, of course, also an interest. And if you compare old paintings, so I didn't put here the slide in, but then you may see that the cereals were taller than the farmers at that time. This was not the reason that these people were smaller than us, but just that the cereals grow much taller at that times. So, and these are not just for the cereals, so these also takes or holds true for all the other crop plants which were domesticated at that time. And I've been involved and want to just include one of the examples where we were involved in a study led by Takao Komatsuda from Japan, where he was interested in this seed shattering. And so here it turned out that two genes are involved in the shattering in barley, which are called BTR1 and BTR2 brittleness genes. And so here you can see on this illustration that these natural mutations occur twice. So we have in the southern Levant a deletion of one base pair, which leads to a non-functional BTR1, while in the northern Levant we have a deletion of 11 base pairs, which leads to the inactivation of the BTR2, which means if one of the two genes is inactive, the grains stay in the spike. And we confirmed this by transgenic methods, where we transformed Golden Promise, which is the model cultivar in barley, which you see here on top. And Golden Promise is mutated in BTR1. And we complemented this with our candidate gene, the functional BTR1 gene. And what you see here on the diagrams on the right is the proportion of brittleness. So this means Golden Promise, of course, is non-brittle. And if you bring in the functional allele, then all the transgenics, so you see here with the plus and the minus, the transgenics became brittle. And this we proved also for the other gene, for the BTR2. So here we had a near isogenic inbred line, which we were able to transform, and here the same holds true. But today I'm talking about using endonucleases, and here the most common one these days is the CRISPR-Cas technology. I do not prepare much slides for introducing this technology, as this is quite common. So the take-home message here is that they are all derived of bacterial immune systems, and there are two components, one which is necessary for the navigation within the genome, and the other one is just a simple scissor, an unspecific scissor. So these two come together and then form this ribonucleoprotein complex and screen for the complementary site within a given genome. So this means you select a 20 nucleotide long fragment in your candidate gene, and this is then the RNA you use to fuse to this Cas protein in order to induce a double-strand break. This RNP will bind to the DNA once it has this protospacer adjacent motif found. It unfolds the double strand, do a conformational change, and then introduce a double strand break, which is indicated here. As double strand breaks are occurring also naturally, cells are prepared to repair this, and there are several ways how such a repair can take place. The easiest one is based on non-homologous end joining. So here, just the two loose ends will be stick together, and as in the majority of cases, this will be without any problem, because otherwise the cell would accumulate too many mutations. But as the process starts from the beginning, once the sequence is properly repaired, so this can only be stopped by mutations, which occur here depending on the Cas enzyme. So here for the most common one, Cas9, it's 3 nucleotides upstream of this protospacer adjacent motif, this PAM. And so here we then either insertions or deletions happen. And as this cannot be predicted, so this is a precision in a way that we know the genomic site where we induce a double-strand break, but the outcome is rather random. So that's why we call this targeted mutagenesis. Gene editing starts for me while using homology-directed repair, but this needs either the sister chromatid in the natural situation, or you can provide a synthetic repair template, which then will be used in order to repair this double-strand break. And if you provide a synthetic repair template, then of course you can also modify the sequence in a way you are interested in. So here, you know the position where you want to change, and also you know the outcome, at least to a certain extent. And this is then real precision mutagenesis. If you look for examples and serials for targeted mutagenesis, so here I have summarized together with Pune Poramini the applications and serials, and this is in an open access book of a very successful cost action about plant gene editing. So you find more chapters, and here you can see that in serials we can find examples where the CRISPR-Cas technology has been used for targeted mutagenesis for traits which belong to abiotic and biotic stress, for agronomic features, for gene discovery, and also for product quality. So you can see that this technology is rather simple for cloning, and that's why it's widespread used for different purposes. As the title of this talk is about precision editing, I want to spend not too much time on targeted mutagenesis, rather on this precision topic. And so here I included some slides of a former project where we wanted to use homology-directed repair in Barley. So you are aware of fluorescent proteins, and here if you look closely, the green fluorescent protein and the yellow fluorescent protein differ only in 2 nucleotides, which make a change here in these 3 amino acids, and this then leads to a different fluorescent outcome. So you can change the GFP into YFP or vice versa. So my former postdoc, Martin Becker, then established a method where we used biolistic in order to transform epidermal leaf cells of barley. And here we use the transgenic barley line, which has a single copy in GFP. So you use then leaf segments and bombarded those with gold particles, which were covered with the ribonucleoprotein complex specific for the GFP and the repair template with a non-functional YFP. And in order to synchronize or to make a comparison, he co-bombarded also a construct for M-cherry in order for normalization, which cells has been transformed and also still survive this treatment. So the idea was that we have initially GFP expressing cells and we co-bombard the M-cherry. So then if the cell express the M-cherry, we assume that some of them will also contain the ribonucleoprotein complex, which are specific for the GFP and also contain this repair template for the YFP. And then if the YFP fluorescence can be detected, this would be a measure for homology-directed repair. So the outcome can look like this, that here we have a cell which was mutated in the GFP, it expresses M-cherry, and also at least one of the two alleles was repaired for the YFP. And if you then quantify this, so here we put the M-cherry expressing cells as 100. So we found that 82% of those M-cherry expressing cells do not have any GFP. So this means we are mutated in the GFP. 35% of those were YFP positive. And then as Barley is diploid, so this means that either one of the two alleles can have the correct repair mechanism and has been used the repair template for the repair. So we have here in total 8% out of this which are poorly YFP, where we assume that here only the HDR is visible, which is a quite high percentage. The problem here is that you have to bring the repair template to the place where you induce the double-strand break. And so that's why here biolistics is in favor over agrobacterium-mediated transformation, as agrobacteria may do not provide sufficient repair templates that this happen. So we did here also transformations in order to get stable transgenic plants, but unfortunately this failed. While we were screening 400 plants, none of them were positive for YFP, as this has no selectable advantage for those cells to survive. What we found in regenerants when we targeted the Barley gene, that here at least 25% of the regenerants had a mutation in the Barley gene. Then, as I say, the problem here is that the repair template might be not in the right place where the double-strand break is occurring. So that's why in 2019, a technology was introduced, prime editing, which sounds very nice because here you overcome this problem that your repair template is not in the right place where the double-strand break is occurring. So what the scientists made, so on one hand, they inactivated one of the two domains so that you do not get the double-strand break, you get just a nick. So they made the Cas9 into a nickase. They extended the guide RNA for the part which do contain, on one hand, the editing sequence and the binding region. And in order to fulfill then the extension of this DNA strand, reverse transcriptase. So you have a fusion protein from Cas and reverse transcriptase, and you have an extended guide RNA, this PEG RNA, which then leads to the desired modification. And so here I ask my master student Jana in order to do this in Barley. We rely here on a system which has been published by the group of Bing Yang, working for multiplex prime editing in rice. And while we tried prime editing earlier, so here I was really convinced that this time, with all the modifications which the prime editing meanwhile got, so by adjusting the guide RNA, the PEG RNA, and as well the Cas protein and the reverse transcriptase, so here a multiplex editing is now efficient. So they used here constructs which do contain multiple elements. So on one hand, we wanted to introduce a mutation in order to make the plant herbicide tolerant. For this, we had to alter the rice gene at 3 amino acids. And at the same time, we introduced then the resistance or tolerance to rice blast by introducing or altering the promoter for the SWEET11 gene. And so here on the right-hand side in this diagram, you see by the green and the gray bars, which illustrate the number of modifications, that you have a high number of regenerants which were altered in the 2 traits. And we also functionally validated this, which is summarized here. So we found plants which were modified, which were then herbicide tolerant, and we were also tolerant for the disease. So by using this system, I then challenged my master student to follow up a strategy to have an easily screenable system, because a master student do not have much time, and I was involved in a study together with Niels Stein, and at that time Ming-Ju Li, who was a PhD student at that time, Niels Stein's group. So where they identified the gene for the famous albostrians mutant in barley, which give this variegated or this striped leaf phenotype. And this turned out to be in the natural mutant a 4 base pair deletion. And so I thought this might be a nice target for prime editing. So what Jana should do, she should use Golden Promise plants, make this prime editing construct in order to get at the end some striped leaves. So as this project unfortunately is not yet fully executed, I can show here only the first promising pictures from the regenerants. So what we find was striped leaves for early regenerants, but what I have not yet ready is the genetic modification. So we need still to amplify and verify that these are really prime editing events. In parallel, in another collaboration, we have confirmed similar efficiencies like Bing Yang in rice. So we also got rice plants which were prime edited at high efficiency. So as barley is quite tolerant for the current climate conditions, so it might be still worth looking beyond cereals. And so here, some of those orphan crops come around, so like quinoa and amaranth. And I want to spend the remaining minutes working out what a PhD student in the lab has recently made, as amaranth is on one hand nutrient rich, contain high protein content, is gluten-free, and also drought tolerant, which might be also relevant for Europe as well. So here I present some data which are part of the thesis of Susanne, and this is a collaboration with Markus Stetter from the University of Cologne. And here the idea or the interest of the both of us was regulatory pathways, as amaranth has been domesticated several times and Markus is interested in the way how de novo domestication happens and how certain pathways can be introduced. And we used Amaranth here as an example because Amaranth do contain also pigments. So pigments are very common in plants. They are on one hand used to attract pollinators, but they can be also used as sunscreen and for several abiotic or biotic stresses. So pigments are quite well studied, as the majority of those pigments are anthocyanins. And here, anthocyanins, the pathway is well known, and also the regulation is very good known. But Amaranth belong to the family of the Caryophyllales. And here you can see this is a very small group of plants which do have developed a different type of pigments, the betalains. So plants which do accumulate betalains do not have anthocyanins. And we were interested in how this can happen. So, as I say, the anthocyanin pathway is quite complex. It has been involved many different enzymes, but the regulation is meanwhile quite well understood. If you look on the betalain pathway, this is indicated here. So there are more or less just 3 enzymes which convert tyrosine, which is present in all eukaryotic cells, into either yellow beta-xanthines or in the red beta-cyanins. So only 3 enzymes are necessary to come here, but the regulation is still unknown. So you might be aware that these betalains, which you may know from red beet, has been also used as a nice reporter system. So scientists fused here the 3 necessary enzymes to make one long transcript, and we included 2 ribosomal skippers in between to ensure that the proteins are separated, can fold properly, and have a functionality. And so here I use this also regularly for my courses here at the university, but of course also for selecting transgenic tissue. So to analyze betalain pathway in Amaranth, Susanne first had to establish a plant transformation technology, as there are a few papers out, but it's not a very common method. So here, Susanne used hypocultural explants and then induced first callus, which were then co-cultured with agrobacteria. She continued callus grow and then wanted to she changed the media composition in order to get regenerance. But here, this in combination with the agrobacterium transformation didn't work. So what we at the end achieved was transgenic calli, but not fully developed transgenic plants so far. So as I say, the betalain pathway contain 3 genes, or 3 enzymes. And in Amaranth, we have for the first reaction, the CYP7682 or 5, while the second reaction is then only for the CYP7682. We have a DODA, and we have here then also a glucosyl transferase, which do then the last step. And Susanne should use targeted mutagenesis in order to inactivate the first gene, this AD2. And for this, she designed 2 guides in the exon 1. So you see here the genomic structure of the 2 CYP genes in Amaranth. So she selected 2 guides for the AD2 and 1 guide for the AD4. And she had another guide just as a control for the Ruby fusion protein. So the outcome was very efficient. So what you can see here also is that she got a high number of up to 30% or 33% of coli which do contain mutations in more than one of the target sites. So she could inactivate these two CYPs very efficiently in Amaranth kali. And if she then looked for the type of mutations, so you typically find this +1. So the 0 means this is wild type. And then if it goes in this direction, this means insertions, while in the other direction means deletions. So here we also find many insertions, but also, as you can see here, deletions. And this more or less is true for all the 3 guides which were analyzed here. So in summary, I hope I could show you that we can use CRISPR technology for functional genomics. So these will lead to the identification of the regulatory networks underlying important traits. So targeted mutagenesis using CRISPR technology was established in barley and grain amaranth. The better understanding of the regulatory networks will help breed crop plants that are more resilient to dynamic climate conditions. And what I also didn't stress here much, but once you know the function of individual genes, you can, of course, combine this and target this by the CRISPR-Cas technology to accelerate de novo domestication, also of orphan grain crops. So with this, I want to highlight here in the acknowledgements the many collaborators which I had over the years. I need to acknowledge the funding sources, which are also numerous, and I need to thank my team. So this is Susanne, which is the PhD student here working on the Amaranth story. And I thank you for your attention, and I would be happy to answer questions, and just want to finish with one announcement that this year we have the International Association of Plant Biotechnology Congress, which takes place this year in summer in Saskatoon, and maybe some people are interested in coming there.
Thanks for the great talk. And there's a lot of different technologies and different biology there. If you have any questions, you can either put it in the chat box, we can read it, or you can virtually raise your hand, and then we can allow you to unmute, and so you can ask your own question. So far, I don't see any questions. Maybe I can start one. I just wonder for the prime editing, what is the size of this DNA fragment that can be replaced?
Yes, so that's a very good point. So here, as this guide RNA cannot be extended, so you are limited to about maybe 20 to 30 nucleotides, which can be used for altering the genomic position. So you cannot introduce a whole gene like with this homology directed repair, as the stability of the RNA would be not sufficient that you can do this larger modification. So this is rather for smaller modifications. Yes, a very good point. Yes.
Another related question is, so what is the efficiency of prime editing compared with a normal CRISPR-Cas to generate an indel or small mutations?
So this is also rather low. So as I cannot give you the actual numbers for barley yet, as here still the genotyping is necessary. So I can just refer to what has been published in rice. So Being Young achieved up to 47% multiplex prime editing in rice. So targeted mutagenesis we have also achieved high efficiencies that almost all the plants do have a modification in the target site. So, but this is for targeted mutagenesis. So this is working very well and rather easy in Barley for this homology-directed repair. So here, for the transient assay, which I showed, we were for this homology-directed repair in the range of 5 to 10%. But for stable plants, we also do not have extensively done this so that I can give you reliable numbers.
Yeah, thank you. There's a question from Dangping Luo. He asked, since GFP and YFP are very similar, how can we distinguish them and then measure the editing efficiency?
So here, of course, you need a fluorescent microscope where you can then just separate the channels, and then you can, as the peaks for GFP and YFP are close to each other, but can be still distinguished. So then you can quantify the individual peaks and can distinguish between GFP and YFP. So this will be not on a regular binocular with fluorescent lamp, but you can do on a confocal microscope.
I don't see more questions so far. So maybe I'll ask another question and wait for others to write their own questions. And so you talk about that this gene editing can be used to de novo domesticate a crop or wild species.
Yes.
So how many genes do you envision you can edit simultaneously? And when you put more guides, more genes, do you envision that the efficiency will drop a lot, or you think it's always high efficiency? And since you work on different species, and do they show any difference, or it's pretty robust for any kind of species you have worked on?
So of course, the efficiency depends also on the genomic location of your target genes. Yeah, so that's why it might be certain areas of the genome which are less easy to access than others. So that's why a multiplex approach will have always a different outcome. So it depends heavily also on your guide RNA selection, but meanwhile there are online tools available which allow you a quite good pre-selection. And I would always recommend to start with at least 2 guides per target, not to repeat the whole transformation experiment if one of the guides does not work. Yeah, so because then you start right from the beginning. So that's why I would use at least 2 guides, and in the best case, you can also cut out between these 2 guides the fragment, and this would help you even identifying those events easily, because if you then amplify your target region, you would have a smaller fragment, and this you can easily spot on an agarose gel. So for multiplexing, I mean, there is this low gluten wheat where we have modified 32 or 36 different genomic locations to get it. So this shows already that targeting multiple locations with a single transformation is possible. There are other examples, like in maize, this breed edit project from Ghent, the BIB. There are some others, like for tomato, we have just made 6 modifications and they converted a wild tomato species into a crop plant. Whereas rice, which was domesticated by just a few SNPs in a given genome, and you can convert a wild species into an elite crop plant. So the limitation might be in the assembly of the construct, but here there are also these different like Moklo systems or tRNA-based genome editing. So here there are theoretically also no limitations in the number of guides you assemble within one tDNA. And the size of the tDNA seems also not that limiting. So as it has been shown in tobacco that you can transfer 100 kb into the genome. And we have also tDNAs which exceed 30 kilobase pairs, and we do not see a difference in the plant transformation in barley. So the tDNA size might be also not the real limitation. So that's why it differ maybe from species to species. And we have also to keep in mind that the model cultivar we are able to transform might not be always the best choice. So that's why using a lead material, typically the efficiency drop. But of course, then you can use the morphogenic genes like baby boom, wuschel, or the GRF GIF in order to improve this. So that's why I think this technology is quite advanced to do multiplex targeted mutagenesis or even multiplex gene editing in crop plants.
It's very inspiring. And we do have more questions. One from Martha Maluk. She asked, is the transformation method for Amaranth genotype dependent? Have you tried it with barley?
So in Amaranth, Susanna also tested different accessions, and at least we can transform different even accessions of Amaranth. So this is possible. And similarly to what we have done in barley, so over the last 25 years, we tested more than 30 different barley cultivars and accessions. And the tDNA transfer is typically not the limitation, because this seems to work quite well, but the bottleneck so far is still the regeneration process. But this might be also overcome, as I just outlined, with the morphogenic genes. And there are many publications coming up these days which show that by using this, you can overcome this genotype dependency. So we have not, so here I'm not sure about the second part. So we haven't used hypocotyl transformation in barley. If this was the question, but we tested different genotypes with different outcomes. So the model cultivar Golden Promise has the highest transformation efficiency. More advanced breeding material can be transformed with a lower efficiency, and we have one EU-funded project, Best Crop, and I've seen here also some of the attendees from where we use Carbona, which is a lead cultivar, and here we have a lower efficiency, but we still get a sufficient number of either mutagenized or plants which overexpress certain genes.
So is this with the help of the morphogenic genes or regular?
No, in barley, it's so I haven't used the morphogenic genes myself in barley yet, but colleagues at the John Innes Center, Shadia Haita and Mark Smedley, they used them as well, and they could not see the same beneficial effect for barley as we find for wheat. So, but maybe as you also have different combinations of GRF and GIF in the different plant species, maybe we need to use the right combination. So that's why I cannot exclude that morphogenic genes in barley would be also of benefit. We haven't done this yet, as we still get sufficient transgenic plants. And so that's why we didn't use it yet.
Okay. So may I just ask, what is the efficiency for the barley transformation in the cultivar, in the elite cultivar you just mentioned?
So in experiments where we used just the GFP construct, we had around 7.5% transformation efficiency. And in the current transformation experiments within the best crop, I think it's even lower. So maybe in the range of 1% in the best case. So here it's really low, but we still get a sufficient number of transgenic plants.
Yeah, yeah. So there's another question from Klaus Pilen. And the comment, nice talk, guys. Why you find much more heterozygous mutations? Not sure for which part. But.
So the heterozygosity, it's a good question. So I would answer the opposite, that the tDNA, of course, will be integrated in natural occurring double-strand breaks. So that's why our primary transgenic plants are just heterozygous. So for the mutations, it's different. So as of course, as long as you have a wild-type allele, this modification can happen. But you have, of course, the guide will just be for the complementary strand. Yeah, so that's why you have only, the target is only on one strand and not on both. And depending on the repair mechanism, we also find from time to time during cell divisions, then this other allele might be also used. And as we do not know what go into the germline, so then it depends on which cell goes into the germline. You have either a heterozygous or a homozygous mutation. I hope this answers your question, Klaus.
So I hope this answers everyone's question. I don't see any more questions so far. Maybe we will conclude today's webinar. And thanks, Scores, again for the webinar. And thanks everyone for the patience, and sorry about the technical issues. We hope to see you next time. Bye-bye.
Yeah, many thanks for the attendees, and also again for the organizers giving me a chance to talk here. Many thanks.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye, thanks.